We're using microsatellites, and microsatellites are simply a repetitive chunk of DNA. So we have a non-repetitive chunk of DNA on either side that we can use to anchor a primer, and we can amplify this repetitive chunk of DNA in the middle. We run that on an automated sequencer, and with some size standards that tell us how big that is, we can then score how big that microsatellite is in those individuals. And we go through and we run this for a whole bunch of individuals. The first step, and this is the part that takes by far the most time, is quality control testing. This is what Allison was talking about in her talk, is that the first thing you do is you go out, you run your DNA, and you find out it doesn't work. And then you troubleshoot over and over again until you get it to work. We've developed a protocol for testing and quality control testing for microsatellites that we're using. And what we do is that we take known crosses of adults, we spawn larvae, and we determine whether or not those microsatellite markers are inherited in the way that we expect. So for example, for Blackfoot Opihi, we started off with 18 microsatellite loci. We go through, we test them. We find that 6 of those loci don't behave in the way that we expect for the basic assumptions of population genetics, that you spawn mom and dad with known genotypes, you score the larvae, and they don't share their genotype with either of their parents. Well, then we don't really trust when you get a different frequency in one population than another. So, or we find things that they have 3 or 4 or 5 alleles in the offspring, and that doesn't really make much sense in terms of standard population genetics either. So after we go through and we cut our markers down to the 12 that we believe and that behave the way that we expect, we then move on. As I said, we started off where we're sort of focusing on Molokai and moving out. The large stars are the ones that I'm going to present the data for. Chris Bird and Adam Baker have been burning the midnight oil to try and get some data for this presentation. Chris has been analyzing the data over the holidays and has data so far for these. Most of the samples that we have been run through the sequencer, but now it takes a lot of time to go through and score them. So I'm going to present some data just for the island of Molokai. So we're looking at either end of the island. And what we find is, in terms of looking at the distribution of the allelic variants, they're identical. For these two populations on either side, we see no structure, significant structure across this island. And we find that on each of the island comparisons that we've done so far for all of the examples. But if we take a look island by island, when we compare one island to the next, for every one of the pairwise comparisons that we've been able to complete so far, every island is significantly different than every other island. So that suggests that there's a fair bit of structure, and in this case, up to 50% divergent between one island and the next, relatively close to one another. So we see big population differences from one island to the next. So in terms of the current conclusions based on the data that we've been able to analyze so far, and the rest of it has still to be analyzed, so I emphasize this is preliminary, but we see currently no population structure within a single island, but significant structure between each of the islands that we've been able to analyze so far. And so if this preliminary result holds, what that suggests is that every main Hawaiian island needs to have its own MPA in order to have any effect on the spawning and the harvest on the other islands. So for example, the reason that Opihi on the island of Oahu may not return is if we've harvested them on this island out of existence, it's going to take a very long time for them to return from any of the other islands that have large populations elsewhere. And so what remains to be done is that we have 3 additional sites to finish the scoring for 11 additional loci for the Blackfoot. And we have the Yellowfoot Opihi. There's 29 microsatellites that we've done. The crosses are there. We have to analyze whether or not they pass the quality controls. And we have yet to go into the koele at all. We have the markers that have been developed, but that data is yet to be done. So in terms of the project, we still have to genotype all of these and to get that stuff scored. So we're working on it. We have certainly, this data will be finished. The idea is that this will certainly be done within the next year, but it's going to take a while to score all of the data that Chris and Adam have generated so far. And we want to thank sort of a cast of dozens of people who have really helped out with this work. Funding from a variety of different sources that has helped with the collections up until this point, the development of the markers, and Hawaii Coral Reef Initiative for funding the microsatellite analysis at this point, and all of the folks who have helped out in terms of permitting and scoring of the data.